Assalamualaikum, Prof.
Salam. Widya, apa kabar?
Sehat, Prof. Belum ada orang nih, Prof. Kita tunggu sebentar ya.
Terlalu pagi, ya. Terlalu pagi.
Masih ada. Sehat, Prof.
Alhamdulillah.
Masih cell culture, banyak cell culture sekarang.
Ya, masih, tapi student tidak ada nih. Itu masalah.
Ya sih, itu udah enggak sih, tapi yang PRGS mah pakai ini ya, pakai apa? Pakai RO kan ya? Masih Putri, bukan?
Masih.
Masih ya?
Mau di extend lagi, belum selesai lagi project itu. PRJS tak boleh pakai untuk student. Dia kan di-tag.
Harus pakai RO ya.
RO, maksudnya dia bukan student lah. Bukan student.
Putri kok udah selesai ya?
Sudah. Mungkin selesai proyek ini, mungkin dia mau cari kerja lain mungkin.
Putri itu orang Malaysia apa orang Indonesia?
Orang Malaysia.
Malaysia. Jarang-jarang orang sini nama Putri.
Betul.
Ada satu tuh student dengan Dr. Denny. Saya ambil satu student.
Proyek
yang extension dari FRGS itu. Gaya-gayaan bikin satu PhD, satu master. Baru
kalau sudah dapat scholarship dari luar boleh? Kalau sudah
memang masih enggak, udah ngambil dari grant. Ambil Dr. Denny satu, ambil saya jadi co. Untuk lebih ke phytochemical study untuk project yang sama.
Yang dekat dentistry program itu spesialis ya? Dokter spesialis.
Iya, yang dokter spesialis, dokter orto. Jadi dia kalau dulu dia istilahnya, kita ada orang. Kita stop dulu kali Pak. Yang master and PhD-nya masih 2022 insya Allah, udah stage terakhir. Yang by research. Try slide dulu, Prof.
Is that okay?
Ya, it's okay.
Stop dulu ya.
Assalamualaikum Pak Ci.
Aku salah Adi. Dan join dari Palembang ini Bu.
Oke. Selamat datang Ibu Ade.
Karena open kan.
Selamat datang.
The more the merrier, Prof.
Terima kasih Bu Widya sudah diizinkan masuk.
Iya, sama-sama Bu.
Karena
dia bagi code password sekali kan, dia tak perlu minta izin kan?
Iya, enggak.
Langsung ya.
Perlu ya?
Enggak perlu admit, langsung aja.
Langsung.
Open
aja tuh masih kurang, Prof, biasanya.
Betul. Karena orang banyak aktivitas juga kan?
Iya, banyak yang ada kerjaan lain. Nggak apa-apa. Kita tunggu 3 menit lagi, Prof.
Oke. Pencahayaan di kamar ini tak begitu jelas lah, gelap nih, muka hitam jadi hitam. Kebetulan hitam juga kan? Tak ada.
Prof, beli satu flashlight itu loh, Prof, yang di depan.
Sudah ada nih. Sudah ada.
Oh ya, sudah ada. Harus ditolong filter lagi kayaknya.
Ada lagi di kampus, di rumah?
Di rumah, Taher, soalnya hujan.
Oh ya.
Jadi WFH, izin.
Yeah.
Oke, Prof, kita mulai saja mungkin ya, Prof.
Boleh.
Okay, alright, okay. Assalamualaikum warahmatullahi wabarakatuh. Thank you very much, everyone, for joining us today in our sixth fast talk for 2021. And today we are very happy to have our speakers, Professor Muhammad Taher bin Bakhtiar. So a brief introduction regarding our speakers today. Dr. Taher actually is obtained his Bachelor of Pharmacy from University of Andalas, Indonesia in 1997, and followed by his Master and PhD from UTM, Johor, okay, in 2000 for master and 2005 for his PhD. And currently, Professor Taher is actually our professor from Kulliyyah of Pharmacy, and his research interest basically is on drug discovery involving extraction, isolation, characterization, and bioactivity study of natural products from the plants. So his current research is more on wound healing, anti-cancer, anti-obesity, and anti-diabetic study for over the several medicine plants using the animal cell culture. Prof. Taher is also published a lot of papers in reputable journals and also contributed in his in the chapters of the book. Okay, without further ado, I would like to invite our speakers today, Prof. Taher, to deliver his talk regarding the anti-diabetic study in the cell culture. Prof, the screen is yours. Thank you.
Thank you very much, Dr. Widya, for giving me an opportunity to give a first talk. Yeah, first here, I'm not sure how many minutes do I have. Just to try my best to make it very light. Yeah, even though the content may be quite heavy, but I'm trying my best to be more friendly, talk to all audience. It is not actually a lecture, just sharing my experience with regard to the use of cell culture in vitro study. So allow me to share my screen. Is it visible?
Yes, Prof. Yeah.
Okay, Bismillahirrahmanirrahim. Alhamdulillahirabbil alamin wassalatu wassalamu ala asrafil anbiya'i wal mursalin wa ala alihi washabbihi ajmain. Abishrah li sadri wa yassir li amri wa hlul 'uqdatam min lisani yafqahu qawli. So thank you very much, Dr. Widya, for giving me an opportunity to share my research with regard to the use of cell culture in anti-diabetic and also obesity research. Actually, this I was started my research on the use of cell culture and anti-diabetic study during my PhD. Yeah, during my PhD. So I use cell culture into my PhD to study anti-diabetic effect of cinnamon or creaminess. So and then until today, I still use this kind of in vitro study, yeah, to check, yeah, the ability of certain compound or drug, or maybe extract and pure compound for the activity in to be used in anti-diabetic. Indeed, actually, anti-diabetes or diabetic is quite complex, yeah. Not just a simple disease. It is actually very complex metabolic disease that can actually very still what call it to be explored, right? When you get to the how to manage it, yeah. Actually, diabetic is actually some theory mentioned that it is reversible, yeah? Meaning we can be cured from diabetic. Yeah? So the title of this talk is anti-diabetic and obesity, because obesity can lead to diabetic, yeah, because of the effect of the fat, excessive fat in human body. They can cause diabetic, yeah. So the phenomena is called as insulin resistance, yeah, making insulin doesn't work properly, yeah. Because in insulin resistance, in fact, the body is still producing insulin, but they are not able to bind the receptor, yeah, in order to allow certain cascade or signaling to uptake the glucose to the cell. Yeah? As we know that actually in diabetic, the cell is starving. Yeah, I mean they are lack of nutrient, especially glucose, for their routine activities. Yeah. So in this case, meaning we have to find some substance or compound that can improve the insulin sensitivity. Yeah. Also to find other substance that can work as insulin. We call it as insulin mimicking agent. Yeah? So with regard to diabetes mellitus, actually we know that actually diabetes is very close to insulin, right? Because insulin actually is the hormone that very important in our body to regulate the use of glucose for energy and other kind of activities. Yeah. So indeed, insulin is very the most actually gift from Allah Subhanahu wa Taala, that actually that one is for normal people, they don't need actually anti-diabetic other than insulin that already produced by our body. So in nature, actually, there is small molecules that can act as insulin. We call it small molecules. Yeah, small molecule here referred to because of the structure is small compared to insulin. Insulin is actually protein is made by peptide by amino acid, yeah, making it to be very large molecules. Yeah, so we call them as macromolecules. Yeah, it is also similar to other type of macromolecule inside our body, like interferon, interleukin is considered as macromolecules. Yeah, and nature from plant, microorganism, and other secondary metabolite, we call it as small molecules. So so in this talk, we focus on small molecule, yeah, to be used to manage glucose in our body. And then we can also refer to type of diabetic. Yeah, there is type 1 and also type 2. Yeah, type 1 is insulin dependent diabetes mellitus. It is very depending to the insulin. Yeah, and type 2 diabetes mellitus is actually non-insulin dependent. Yeah, so it can cause by insulin resistance. Resistant means is inability of insulin to works properly, yeah, to uptake glucose that available in the blood. Yeah. So as I mentioned just now, that actually the diabetic cells, diabetic cells actually starving. They need actually glucose, but the cell is not able to uptake, yeah, to take the glucose to be utilized for energy and other kind of metabolism. There are many actually method or experimental way actually how to study anti-diabetic drugs. Yeah, it can be done using enzyme activities, as it is quite popular method. Yeah, using alpha glucosidase, beta amylase, yeah, to check the activity of the enzyme that involved in glucose metabolism. Second, using in vitro study is using cell lines. Yeah, mainly we use the cell that has insulin receptor. Yeah, a good number of insulin receptors like adipocyte cell and also myotube cells. Yeah, adipocyte cell is actually a fat cell. It is very good to be used in anti-diabetic study because this cell has many insulin receptors. Yeah, so it will be good to check the substance can be used for anti-diabetics purpose. And also we can use in vivo, yeah, using animals, animal study using mice or rats. Yeah. In the study, normally we induce, yeah, animal to be diabetic using streptozotocin or alloxan. Yeah. And also in vivo study, we can also studying glucose uptake by isolating, yeah, isolating the tissue or cell from animal. The last one is clinical study. Of course, this one is much quite advanced, yeah, meaning that that is already formulated and has been studied for the toxicology, whatever. Yeah, so it is using human in the study. Based on the research practice, yeah, why we need to do in vitro study first before using animal. Yeah, because in animal study, the principle, yeah, because we need to replace, whether the study can be replaced with other method, yeah, and so to reduce the number of animals and also to refine, yeah, meaning less is better, yeah. So before we move to animal study, normally in ethical committee meeting to approve our proposal to use animal, normally they will ask whether you have in vitro study, yeah, proven in vitro study to be proceeded with animal study. So we so that with that what called the requirement in vitro study becoming and actually the preliminary way actually to establish the activity. Okay, diabetes is considered as non-communicable disease. It is not spread to other people. Yeah, mainly it is maybe through genetic, inheritance. So this one of the most disease, yeah, for just in 2016, yeah, like cardiovascular cancer and so chronic respiratory. In fact, diabetes with regard to the projection, yeah, so it can affect 578 million people in 2030. Yeah, so the trend is increasing. Yeah, perhaps because of lifestyle changing. Yeah, and also maybe because of a free radical, yeah, because radical exposure of our body, yeah, toward the food intake and maybe through environmental condition, yeah, that making inflammation to the cell, and then it has killed about 1.6 million people, and based on the record, yeah, 1 in 3 adults overweight. Yeah, overweight means it is obese. Yeah, obese obesity actually can lead to the diabetic. Yeah. So if we see the expenditure, yeah, for total diabetes-related health expenditure, because we know that diabetes is what call one of the source of sustained kind of disease. Yeah, I mean a trigger to other type of disease like cardiovascular. Yeah, retinopathy disease and many other diseases. Yeah, so for total expenditure, according to IIUM, in 2020, yeah, in 2019 it is about 760 billion USD been spent to manage, yeah, or to treat diabetes related disease. Yeah, and it is projected that into 2045 about 840 billion US dollar, yeah, will be spent for this kind of disease. So I think need to do something at least in what call the academic or university level to find something that can actually help this kind of phenomena. Okay, obesity and diabetes it is actually very close. Yeah, it's believed that 80 to 85% of the risk of developing type 2 diabetes, yeah, is caused by obesity. Yeah, so this because of the theory that the fat, yeah, can cause insulin resistance. Yeah. Actually, fat can affect the sensitivity of insulin receptor. So yeah, so making cells not able to uptake, yeah, to uptake the glucose to be used in our body. And with regard to the statistic as well, about 80 times, yeah, obese people will be diabetic. Yeah. And this actually has been released with regard to the obesity number of the population in certain countries. We have the record for that. Yeah, so meaning by increasing the number of obesity in one country, automatically will also increase the diabetes patient. So small molecules have been used to treat diabetes, like these oral antibiotic drugs nowadays that are available in the market. Yeah, they have several groups, yeah, and several mechanisms, and like sulfonylureas, yeah, it acts to stimulate pancreas to produce more insulin. There's another class, yeah, mellitus, yeah, stimulate that one, yeah, and also thiazolidinediones, TZD, actually it is increased insulin sensitivity. So meaning when we are trying to search anti-diabetic drug, we have to understand first about the mechanism action that can be proposed. Yeah. So how to do that? Yeah. We can do cell culture. Yeah. We can do cell study. Yeah. In fact, according to pharmacopoeia, many of drug they will the many drugs explain the explanation of the mechanism is through cell, yeah, because cell is the smallest unit, yeah, in even though actually the DNA is the smallest, but the cell is one call it the one individual. Yeah, this they have organelle that also responsible for to give a response to the certain treatment. So so the use of cell culture actually to explain the mechanism of action of the substance in order to be accepted, yeah, for further study. Yeah. If we refer to drug discovery and development chart, yeah, when we try to find a new substance to be developed to be a new drug, yeah. So the level of study in vitro, yeah, that we are going to propose, yeah. So it will be here using in vitro study, part of the screening activities, yeah, to check whether the substance is potent to be developed or to be proceeded with next further study, yeah, because this process is quite lengthy. It is about 12 years, and also cost can be $1 billion USD, yeah, and the last one the approval just only one later on, yeah. Just it is very strict, yeah, very strict. So our study for in vitro is at this level, yeah, doing screening for their activities. So there are some type of cell that can be used in anti-diabetic research. Yeah? The first one is adipocyte cell, yeah, or cell myotubes, islet cell line, yeah, hepatoma cell. So all of them are giving a response, yeah, toward the treatment with the glucose. Yeah? Of course, we need to start starving, yeah, to do starving the cell first before we treat with the compound or with glucose. So this the common cell line that can be used in anti-diabetic study, yeah, using in vitro technique. So we get to the morphology, yeah, like this one is adipocyte cell, yeah, and C2C12 cells, yeah, RIN5F and HEP2G cells, and they have different morphology. Yeah, this one is fibroblast-like, and down here is like epithelial-like cells. Yeah, so they have different morphology. Yeah. So because we are studying at cells level, yeah? So we have to understand actually how glucose is utilized by our body. Yeah? Actually, the process is quite complex, involving several cascade or signaling and also location of certain protein and gene. Yeah. So let's say this one is a cell. Yeah. And this receptor here, insulin receptor around here, yeah, surrounding the cells, and how the glucose is uptake, of course, through signaling. Yeah, the best explanation is using insulin. Yeah, insulin will trigger, I mean, to stimulate the insulin receptor to open the gate. Yeah, and certain and also conducting what call it other signaling for translocation of certain protein and also gene. Yeah. And also to activate glucose transporter, yeah, to transport glucose into the cells. So what happened in the patient who are suffering with insulin resistant? So in this cell membrane, maybe something not correct happened here. So perhaps insulin receptor is not really sensitive toward insulin. And also perhaps the number of insulin that is produced by pancreatic beta cell is not sufficient enough to give what called to stimulate to respond to the glucose and the bloodstream. So in the cell study using pre-adipocyte sample or adipocyte cells, so we have to check whether the substance that we are going to test responsible to insulin receptor activation or stimulating several signaling or cascade. So in our study in vitro, so we need to check to prove that substance is actually working to utilize glucose, whether the amount of glucose is uptake in good number. So we can check using in vitro study. So as control in vitro study, normally we use insulin and other small molecules like rosiglutazone, or maybe we can use metformin as well to check whether the substance is work similar with insulin or can be used to replace insulin. So in when you get to glucose uptake, the possibility of the drug that we use actually can be mimic. It can mimic insulin action, and also it can sensitize insulin receptor. It can increase the sensitivity of insulin receptor to bind to insulin, especially for the treatment of insulin-resistant diabetic patient or type 2 diabetic patient. So their problem is around here, around the cell surface, cell surface, toward the response to the glucose. And in vitro study, we can make cell to be insulin resistant. We can induce cells to be insulin resistant using PAL method. So this is really the data. How actually we can do in vitro study. We can make the cell to be resistant. Try to treat with our substance, and then of course we need to use control, positive or negative control. So whether, and then we check for the activity for glucose uptake, after we treat with our substance, and then we can check whether the substance able to relieve the cell from insulin resistance, meaning it can activate insulin receptor to be to allow the binding with insulin. Okay. So in our daily life, there are many plants that have been reported to have anti-diabetic action. Whether that one is with scientific evidence, sometimes not, sometimes. So actually that one that is our responsibility actually to answer the people, because the people mentioned this plant is good for antibiotic, but we are not doing anything actually to prove it. Actually not really good practice for our as actually has capability and also has facility to prove that claim. Based on record, about 800 plants have been reported to have this anti-diabetic activities. like banaba plants, is Largosemia, Lagestromia speciosa, yeah. Bittermelon is Mamordica carantia, is called is Peria, yeah. Peria is actually, but until now, there is no clear explanation with the what activity of this Mamordica carantia, yeah. Yes, our member here, Dr. Alfi Khatib, is doing research on Mamordica carantia on antidiabetic, yeah. So using zebrafish, yeah, zebrafish for antidiabetic model as well, yeah. And also it also reported for Chinese ginseng, yeah, Panang ginseng to have this antidiabetic activity as well, and also cashew leaves, yeah, Anarcadium occidental. I think my collaborator, Dr. Hanapi from Kulliyyah Allied Health Science is doing this research. Yeah, cinnamon. Yeah, it was my PhD. Yeah, and clove, fenugreek. Yeah, genema, and also Garcinia atroviridis. Yeah. The interesting fact for Garcinia atroviridis, yeah, it is also being used to to reduce fat or for anti-obesity. Yeah, if you search from internet, you can see some commercial product. Yeah, has been marketed. Yeah, have been marketed for Garcinia atroviridis, yeah, or some glugel, yeah, some cupping. Yeah, okay. Fact about the Garcinia atroviridis, yeah, it is containing active component. We call it hydroxytric acid, yeah, or HCA, yeah, to be used in weight management, and also HCA is also main acid in the fruits. Yeah, if you try to test, asam peping or asam glucose, yeah, it is very acid. I call it asam, yeah, and so based on this, yeah, fact, we try to study. Yeah, so after this, I will share my our research finding with regard to using cell culture, yeah, in antidiabetic or anti-obesity obese study. So this Garcinia atroviridis, yeah. Some cupping or some glugo, yeah. This the product, yeah. Actually the many brands, yeah, in the market available to be used for fat reduction, yeah, diet, yeah, to be used for anti-obesity. So the flow of the study. Flow of the study, yeah. The first, what we need, yeah, in natural product study using as a line, yeah, or in vitro. Of course, we need to do extraction. Yeah, we need to extract the active component using organic solvent. Yeah, and we'll do phytochemical analysis. Yeah, to check what is the component. Yeah, what is the component in the extract. Then we do in vitro study, and we need to establish which parameter to be observed, yeah, in our study, yeah, like cell viability assay, yeah, adipogenesis assay, leptin secretion assay, also adipocyte breakdown, and also Western blot. Western blot actually is to check the protein expression after we treat the cell with certain substance. So this is the process. We extract using water, and then we dry it using a freeze dryer, yeah, to get the dry extract, yeah, the dry extract. So the product that is available on the market, they just pack this, yeah, they formulate this freeze dried extract, freeze dried extract, and formulate it in capsules and sell it to the people, yeah. So they claim that it is effective, yeah, to be used in obese, or as anti-obesity. Then the good practice in the natural product, when we are trying to to test the extract to the cell, or to the animal as well, we need to do phytochemical analysis, to check the presence of our target compound. In this case, our target is hydroxycitric acid. So we need to check the amount, because later in our discussion we should explain, what is the role of the active ingredient in our study. Then in vitro study, I think this one is quite common, but maybe it is also important to share here. So in vitro study using animal, we need because cell culture is sensitive. It is very sensitive to chemicals. So we need to use the concentration, or the dose that safe to the cells. So we need to check the IC50, check the C50 of the substance, check the cell viability. After that we can generate IC50. So normally the dose that we use in the experiment should be below IC50. So the concentration that doesn't kill the cell. So of course we need to do serial dilution to find out the IC50 of the compound or extract. Interestingly, adipocyte cell, when the first time we culture, the cell is not matured. It is in the form of pre-adipocyte. So to make it matured, we need to do several induction process to pre-adipocyte cell to make them adipocyte. It will take about 14 days to check the cell is matured. So during this process, we can do several observation. Several observation, also checking. So to check our compound is mimicking insulin or not, we can replace here. Because during the induction we use cocktail of MDI cocktail containing dexamethasone, IBMX, and also insulin for a few days. And then we can replace insulin with our compound if you want to check the ability of the compound to mimic insulin action. Until it is the cell is matured. So the matured cell, it will be noted with the growth of fat inside the cell. So this example of our study, adipocyte differentiation. So they use MDI is containing dexamethasone, insulin, also IBMX, containing Garcinia atroviridis extract, 10 microgram. And this one 45, just to compare the different doses. And also this untreated. You see untreated, there is no fat accumulation. So in the adipocyte cells, the formation of fat inside the cell, it is like droplet inside the cell that can be stained using oil red O. We call it oil red O. If the cells is already converted into mature adipocyte, so the formation of lipid inside the cell, fat inside the cell will be a maximum level. This one for control. So what we can say here, just to give you an overview regarding this study. So when we treat the cell with Garcinia atroviridis extract at 60 microgram per ml, so there is no, less, lipid droplet formation, meaning the claim, just not really what call it the final recommendation yet. So meaning that it is true, the Garcinia atroviridis extract can inhibit lipid formation, so it can act as anti-obesity, thing like that, This is untreated, there is nothing happen here, and this one is control. Control is actually the original, I mean the standard hydro citric acid, to make a lipid droplet, So meaning to say that this one of the evidence, to show the substance or extract can inhibit lipid formation, in the cell. And also this is another example, of a different study, It was done by Sister Wastuti 2012 with regard to the inhibition of lipid formation, of adipocyte in our study, You see this undifferentiated control, and I have seen there is no, actually to conclude that there is no inhibition, of lipid formation. So this evidence or this result actually can support that maybe perhaps the extract is good or active to be used as anti-diabetic or mimicking insulin action. Okay. So in the finding, in the result of the use of adipocyte in the study, whether the result will lead us to anti-diabetic effect or anti-obesity effect. If the formation of lipid droplet, meaning the extract is stimulate the glucose uptake by the cell, and then the cell is able to make this lipid inside the cell. And then the amount of actually we are talking to be what scientific talk actually, not perhaps the layman not really understand with this. So the previous one was actually qualitative, and also we can do quantitative, actually to see how much lipid has been formed in the experiment. So we can also extract the lipid, and we read the solution with UV spectral, to check the amount of lipid. I mean to compare the amount of lipid formation within the tested extract. So we can check which one is the highest, which one is the lowest. Okay, now after that we can actually present our result with the picture, qualitatively and also quantitatively, So we can this we can be captured using microscope, and we can also quantify the amount of lipid, and then we can convert into the chart. In our study, we also check for the formation of adipocyte, adipocyte using this Adipogenesis kit, So what happened? Actually, 60 concentration actually exhibit the low activities, to make to stimulate adipocyte formation. So we study adipolysis, and then Another experiment to prove the activity of the extract as anti-diabetic is on glucose uptake activities. So how much glucose will be uptake by the cells, we can quantify. This example from this Yamamoto 2015. They use control insulin and use the compound. So glucose uptake can be quantified. This control is untreated and is insulin, and it is the compound. And also the experiment of glucose uptake can be conducted using a radio label. This one is radio label glucose. So we can use radio label glucose. The amount of glucose that we consume by the cell is very little, very little. Sometimes we cannot quantify it using just a normal chemical reaction because it is very small. So if we use radio label glucose, no matter how small is it, so we by using liquid scintillating counter, actually can read the quantity of the glucose that has been uptake by the cell. This one is using non-radiolabel. We call it 2NBQ reagent. Actually using a fluorescent glucose. Fluorescent glucose. When the cells uptake the glucose, actually it is fluorescent. We can observe using microscope. We can check, the amount of glucose that have been uptake by the cell. This is an example. So we'll get this result. So we'll compare between our samples, and later we can discuss about the result in our discussion. So this is an example of the glucose uptake using red fluorescent glucose. See, we use microscope, fluorescent microscope, to read our cells under this fluorescent condition. So the amount of the glucose that uptake by the cell, we can observe using microscope. You see this, the number is high. This insulin is specific control, and then the less, maybe this the MSO, maybe this one is control, negative control. So this qualitative observation, and this glucose uptake also can be quantified using spectral. So we can present the data using this bar chart. And then with regard to glucose regulation, it involves first of all cascade, and also signaling and expression of certain protein as well as gene. So in this study, actually we can check the protein involved in the glucose uptake cascade or process, like PPR, gamma. We can do it. We can check it. After we treat the cells with the compound, and then the cell will be collected and extracted, and then we can run Western blot to check certain protein expression. Because the higher expression of certain protein target, meaning it can help the consumption of glucose. So, for example, for this, PPR gamma expression, you can check using Western blot. Of course, the process is quite lengthy process, and must be done in what I call the quite experienced researcher to check the formation of this protein expression. And This expression, protein expression also can be displayed, presented in figure and also visual, or can also be quantified using gel-doc system, gel-doc system, gel documentation reader system that can convert the spot to be a number, and the number can be presented as this bar. Other protein expression in glucose utilization process in the cells is this CBP alpha expression. So similar work to PPR gamma just now, we use Western blotting as well. Western blotting to check the expression of this protein. And it's very technical. So this kind of protein that can be observed. And other than the expression of protein, and glucose utilization by the cell involves gene expression, because gene is related to protein, right? Increase the gene expression, of course, the protein also will be increased, because gene is related to DNA. DNA is responsible to make protein, certain protein. So we can also check, to confirm the finding, by using RT-PCR to check the amount of gene expression, to check the amount of gene expression, whether it is the gene that we target is there or not, because that one is very important, to support our finding in glucose uptake, So later we can collect the information, and then we can propose the mechanism. In our study, we also study leptin hormone expression, of the cell after we treat with Garcinia atroviridis. So leptin is responsible for food intake. So if it is less, meaning less, if it is high, food intake will be less. So this is actually the parameter that can be observed in this study for anti-diabetic and also anti-obesity study. And then we collect all information, with regard to our experiment, and later we can propose. Of course, in this study we have to use positive control. You have to choose our own positive control, like a small molecule that has been used in insulin management, like metformin or rosiglitazone, to support our finding. So later we can propose the mechanism of action of our substance. So for conclusion, cell culture is in vitro technique which provide proposed mechanism at the cellular level with regard to the action of anti-diabetic and also anti-obesity drugs. Of course, further testing is required to prove the activities using in vivo, and also we need to study toxicity level of the compound and efficacy. So we have done some research collaboration with other researchers in IIUM, and also other university and also the country. So thank you very much for that collaboration to everyone here in this list. And with that, I end it with thank you very much for your attention, and I will return it back to Dr. Widya. Thank you very much, Dr. Widya.
Okay, thank you very much, Prof. Taher. It's very interesting, and actually it's gained a lot of knowledge from this talk. Okay, we open for discussions. Any question from the members for a more clarification, or any kind of question, it can be passed to Prof. Taher. Prof, maybe one question from me first, Prof. Regarding the efficacy study, the extension of this efficacy, it must reach up to this protein level, Prof, or it just like the Western blotting and RT-PCR is a must to do this analysis.
Yes, if we want to develop the drug, to be, the substance to be a drug, we have to conduct that kind of experiment to prove our, to do comprehensive study, and also to get all required evidence to support our finding. Otherwise, we cannot move to further step, because we don't have enough evidence for that activities.
Okay. All right. Thank you, Prof. Any question
but
from
normally
the
we
members?
just stuck because of some time for Western blot. Very costly. So
This is my concern actually, Prof. For antibody actually, right, Prof? Is really expensive. So whether that we can skip or it's a must to do this.
For publication, because we are talking in academic environment. So for Q1, Q2, normally they
require this one.
And if we provide that data, sometimes they will appreciate a lot with our finding. Perhaps the paper will be accepted.
Okay. All right. Thank you, Prof. Okay. Anyone else from the members to raise the question? I have seen there's a lot of people here. Anyone else? Dr. Besma? I just.
Assalamualaikum.
Yeah.
Thank you, Prof. Taher. A really interesting presentation and very good results and a very nice topic to discuss about. I'm really interested in leptin hormone, and I worked for a long period on this. May I ask you, Prof, if you test also the leptin gene and the leptin receptor in this, only the hormone itself?
Thank you very much, Dr. Muhammad. You just check the amount of leptin that is expressed by the cell to the supernatant. We don't check for gene and protein. Yes.
Yeah, because as you know, Prof, that leptin hormone secreted from different types of cells is not very specific. Any adipose tissue can produce the leptin hormone, but the receptors in the genes sometimes is located in certain places. So maybe for further collaboration, maybe, Prof, we can work hard on this. Okay, inshallah. It's very interesting. Thank you so much, Prof, for the nice presentation. Thank you, Dr. Widya, for arranging this. Thank you.
Thank you, Dr. Besma. Prof, we have one question actually from the chat box from Sister Dalila UTM. I'm Dalila from UTM. Is there any option other than Western blot and RT-PCR?
Actually, option here, because to check the expression of the protein, protein is considered as what called antigen. So we need to check it using Western blot, because to check the binding antigen with the target antibody. And also PCR is to check the gene expression. I think there is no option to, so far that as far as I know, to give more evidence to the activity of the drug to be used in antidiabetic other than RT-PCR and Western blot. Until now, the people are still providing that kind of data to present their finding to support the activity of the drug or substance.
Okay. Thank you, Prof. Any more questions from the members? So I think it's no more, Prof. The question. I think thank you very much, Prof, for your time and your excellent sharing knowledge to us. So hopefully we open for collaboration. So anyone interested to collaborate with the Prof Taher, I think is most welcome. So hopefully in the future we can have more collaboration on this anti-diabetic studies. Thank you, Prof. If no more question, yeah, we close session with tasbih surah kifayah and surah An-Nas. All right, thank you everyone. See you again next month. Okay. Assalamualaikum warahmatullahi wabarakatuh. Thank you.
Thank you very much. Bye.